Oh, thank you, Shiloh. I'm not actually doing any interviewing. I'm just doing the setup. I warm up the crowd for the star. Speaking of which, my really expensive cheap watch says it's right on 2 o'clock where I live, or 1 o'clock SLT. And so I would like to begin. And I want to begin by welcoming everybody here today. This makes Friday afternoon so special when we have a group of people like this and an expert presenter like we have today. My name is Meryl McBride, and I have the pleasure of serving as your host today. I want to welcome you to this installment of our VWEC Expert Series, in which we ask educational professionals to share their experiences and their insights into ways that virtual worlds can be used to engage students. I am pleased to introduce today's guest speaker, Marianne Clark, or known more affectionately as Max Chatnoir, who will share with us how she uses Genome Island for teaching and learning. Marianne taught biology for roughly 50 years, that's half a century, mostly at Texas Wesleyan University, before she retired in 2022. She was looking for a place as immersive as World of Warcraft where she could teach biology online when she stumbled on Second Life. Her relative inexperience with scripting notwithstanding, she was able to create a teaching space in SL for biology non-majors, a class that she first taught in 2007. She began with a parcel on the SL mainland, but eventually relocated to Genome Island, which at the time was a part of the Skylands. Her mission, apart from teaching biology, was to share her vision with colleagues and others on the value of social virtual worlds to the teacher's craft. She has a PhD in biology from Bryn Mawr College, awarded in 1972. We will be using Speakeasy for Marianne's talk. In addition, please know that this presentation will be recorded and streamed on YouTube. You can find that stream at the address that we will insert shortly. Feel free to post questions to chat during her brief talk, and Marie and I will collect them for her to answer after her presentation. Now, it's my privilege to turn the mic over to Marianne Clark.
I almost forgot how to stand up. Thank you so much, Meryl. I really appreciate being here. And I guess I will just get on with it. Oh, by the way, the dog in the picture you will probably hear at some point during this presentation. I want to start by disclaiming expert status. Sorry. I'm not an expert, but I am a very enthusiastic explorer, and I like to learn new things. And failure doesn't scare me too much. The thing I really love about Second Life is its immersiveness. What did I learn? I learn best from explanations in text, but I remember in pictures. So Genome Island is sort of a little visual tour through my brain, a representation of my understanding of genetics, which is backed up with text explanations. As Meryl told you, I got into Second Life after spending a summer many years ago playing World of Warcraft with my husband. And the power of that game as a learning environment really impressed me. In fact, one of the things that impressed me was that as I learned to find my way around World of Warcraft, I got better at navigating in real life. So it's really quite an effective teaching environment. When a friend told us about Second Life, where you could actually build what you want, we tried it right away. I want to review the history of Genome through one of the first objects that I made in Second Life, which was a big cell. The cell model was one of the first structures that I ever made on my original mainland sim on Immaculate in about 2004. And when I ran out of prim space on Immaculate, about 2007, I moved up to a whole island, which I called Genome. And I placed Genome with a group of other science-related islands called the Scilands. I'll show you a picture of that a little bit later. Genome was mostly genetics, and I, because the cell wasn't quite genetics, I made another island called Cell Island, to which I moved the cell. And that was about 2008, I think. And then later, mostly because of the threat to remove the educational discount, um, I moved the stuff that was on Cell Island to a platform on Genome. There we go. I think I'm caught up now. So when I copied my Genome objects over into OpenSim in about 2014, I spread it out again over 4 different islands in the Vibe group, Mendel, Crick, Huxley, and Hook. And Hook now has all of the cell stuff. That whole group of islands is now hosted by the Science Center, and I appreciate them doing that. Sorry, I'm trying to find, there it is. Okay. Genome is sort of structured like a virtual lab in which students can run experiments to test hypotheses about genetics. And there are several sections: Mendel's Abbey and Gardens, the Tower, Bioinformatics Platform, Population Genetics, Cell Platform, the Genome Galleries, and a Student Project Terrace. There isn't a fixed pathway for moving through the activities, although there are teleports between related activities, like the chromosome comparisons and the bioinformatics platform. Note cards associated with the objects give information about that object or instructions on how to do the activities. Instructors use the island can set their own sequence for the activities. At the landing point on Genome, there is a guide to the activities on Genome. It's this document on the left called Genome Island, a user's guide. And you can get that at the landing point. You can also get it in several other places around the island. Some examples about how I organize the course modules for my own non-major genetics class are in that guide to Genome. And you're welcome to look at any of those. I want to tell you a little bit about the resources on the island. And these include informational displays, interactive objects, several different meeting sites, a student project terrace, and different kinds of quizzers. And you can see all of those in the Abbey, or you can see examples of all of those in the Abbey and the Gardens. In the Abbey, there is a large conference room and a bunch of posters that explain things. So there's information in the Abbey. There's also a meeting site. Um, the greenhouse and the dihybrid garden are two examples of interactive objects. There's a quizzer over by the greenhouse. There's also one of the stations in the scavenger hunt that I use as an orientation. And you can see just to the left of the gateway into the garden, there's a teleport panel, and there are several of these around the island. The next slide shows a bunch of students at the conference table in the Abbey. As you can see, the table's biggish, and you've probably seen the tables that expand, and this is one of them. It will expand to hold about 40 people before it goes through the walls. So that's handy. There are some other meeting places around the island. There is a science lecture theater, which also holds about 40 people. The gazebo, there's one of several, there are 3 gazebos on the island, and one of them is by the Mixalamas, and that seats maybe half a dozen students. Those tables also expand. And then there is a meeting place on the terrace of the tower. And all of these are out of chat range of one another, so that you can actually, several different classes can actually meet at the same time, or at least several small groups of students can meet at the same time on Genome. There's also a project terrace where the students can mount posters or short slideshows. There are about 26 stations at ground level, I think 12 down here and 14 in the back. And then there's another 14 additional posters up above that back section. So you can get quite a few, well, that's 40 again. You can get 40 posters in this area. You can see some students in the project terrace doing their presentations. And for quite a while, we used to do these live and just have people meet at some particular time to discuss one or more of the posters. But during COVID, I moved the live presentations. Well, I didn't move the presentations, but I moved the question and answers to an online discussion board. And that actually worked pretty well. The posters are displayed until the next set goes up, and since I won't be teaching the class again, the posters that are there now will be there until somebody else needs them. In fact, a set of posters that Ellie put up are on that back section right now. So let me just get caught up with this thing. The next few slides that I want to talk about are primarily for objects or displays that are informational. And this one is about the evidence that led to the Watson and Crick model of DNA structure. On that little table next to the left of the double helix is a copy of the Watson and Crick paper. It's only a couple of pages long, so the whole thing is actually there on the table. The students can flip it over and read both sides of it. Or there's also a link that goes out to a print of that paper. The next slide is the chromosome gallery. And this is also primarily informational. Come on, move. There you go. Each of the chromosomes, so these chromosomes are all to scale. At least all of the nuclear chromosomes are to scale. The mitochondrial genome, of course, is not. to the same scale as the others. But these chromosomes are to scale. So the big chromosomes are the ones with the low numbers, and the little chromosomes are the one with the higher numbers, which is the way the human chromosomes are numbered. Each of the chromosomes, if you click on it, will tell you something about itself and how big it is and how many genes it has, and something about a specific gene that is somewhere on the chromosome. And those arrows that you see there show where that particular gene is. In the next slide are a couple of the ribosomes from ribosome row, and this explains how proteins get synthesized on the ribosomes. It also explains the respective roles of messenger RNA and transfer RNA. The transfer RNA brings the amino acids to the ribosomes, and then the amino acids are transferred from one tRNA to the next to string the amino acids together into proteins. The next slide shows one of the genome walkways. And this is a section of the SIM which has information about many of the genomes that have been sequenced. Of course, by now they've overcome my ability to put them all up. But there are representative model organisms, both plants and animals, on this walkway. And also on the west part of the walkway, which you can see in the picture to the left, there are some of the organellar genomes, ribosomes, and chloroplasts. Each of these posters will give you a picture of the karyotype, the chromosomes, that belong to that individual, and also a link to the paper that describes the genome. And all of those genome publications are public access. I think they still are. I think every time they publish a new genome, anybody can read that paper, which is nice. So the next slide is something that I put up a year or so ago about the difference between the influenza and the COVID virus, because somebody was talking about how COVID was just like influenza, and no, it is not just like influenza. It's quite a different kind of virus. So I put that up just for information about those two viruses. That's in the tower on one of the upper floors. This next slide is one of the interactive displays on Genome. And this is about the monohybrid cross, which is a cross between two individuals that differ in one particular trait. And this shows the first 3 generations from those pure breeding parents. And right in the middle of the posters on the left, you can see a yellow pea. And this is what told Mendel that some genes were dominant to others, because all of the progeny of green and yellow peas are yellow. And then in the dish below the poster, you see the next generation that you get by crossing the yellow peas to each other. And that was what told Mendel that the genes had to come in pairs, because only if they came in pairs could you get the reappearance of the green trait in the next generation. And then the next generation tells you that you can have yellow peas that might or might not be hiding a green gene. You can click on each of the yellow peas and see which ones those are. So this follows the rules of genetics, but each time this is run, you get a slightly different set of offspring in those dishes, so that each student gets a separate data set, and that data set goes into the chat record so that it can be copied from the chat record into spreadsheets, and the spreadsheets are also provided. The next slide is of the dihybrid garden, and this is 4 different data sets from the dihybrid garden. And in a dihybrid, the predicted ratio of progeny with each of the possible sets of characters is 9 to 3 to 3 to 1. And you'll notice looking at these that none of these show exactly a 9 to 3 to 3 to 1 ratio. But if you add all 4 of those progeny sets up, you get very close to an exact 9 to 3 to 3 to 1. Not quite there, but very close to it. And what this does is tell the students that looking at a particular population, especially a small population, you may not see exactly what you predict. And you're going to see something different every time. But if you look at a lot of progeny, you will see something very close to the predicted progeny ratios. Oops, I didn't mean to do that. Okay, the mixollamas are a set of individuals. Sorry, let me catch up with myself here. Okay, and you can see how much variability you get within progeny when a lot of different when you have a lot of different possible combinations of different traits. So in this particular example, you have 6 different body regions that can change color. There are 2 possible colors for each of them to be. And the sticks that are next to the mixollamas record the number of clicks it takes to get to any particular combination. And I usually recommend that students go to get them all the same color because that's the easiest one to spot. But they would find the same results if they looked for any particular given population. Oh, it should take, since there are 6 different possible body regions that you're looking at, it should take about 2 to the 6 or 64 tries to get any particular combination of colors. And as you can see, well, you actually probably can't read those numbers, but the first stick, the long one, shows about 100 tries, and the second stick shows 10. And if you take the average of just those 2, you get 55. So it's not quite 64, but it's pretty close to it. And if you do it several times, you will get an average of about 64 possible different combinations. The next of the interactive sets that you see here is the category. And there are a bunch of different regions in the category. The middle one that you're looking at illustrates the inheritance of X-linked traits. And these are different from other kinds of inheritance because males only get 1 X chromosome. So the male chromosomes aren't paired, but the female chromosomes are. So the females are going to follow the rules, but the males aren't. So you're going to get very different kinds of progeny than you would if the genes were on the other chromosomes. The category also illustrates more typical Mendelian ratios and also lethal genes, which also give you funny numbers. You also see some non-Mendelian ratios in the North Garden.
Call from Slidell TX.
And one of the things you see, or actually don't quite see in this picture, is linked genes, which are just in that little garden that you just see a little tiny piece of way over on the left side. On the right, you have intermediate dominance, which Mendel did not report. So these are sometimes called non-Mendelian inheritance. They do follow the Mendelian laws, but the relationship between the genes and the trait are a little bit more complicated. And this is the aviary, which gives you another kind of variation, which shows you what happens if you've got multiple genes that are all affecting the same trait. Feather color in budgies, in this case, you have either blue or yellow or combinations of it or green, or you don't get either one, you get white. And you click on one of these nests to get a set of the progeny, and you can use the progeny, which are flying above the parents there, to infer the genotypes of the parents. Come on. And then this next interactive set is a 3-point test cross for Drosophila. And if you cross dominant recessives for 3 different traits in Drosophila, and they're on the same chromosome pairs, you can use the progeny numbers to map the genes on the chromosome to tell about what the approximate distance between those genes is. So if you click on each of these possible progeny sets, it will give you a number. And each set of 9 will give you slightly different numbers. So again, whichever student do it, is 2 students doing the same exercise are not going to get exactly the same numbers. And I just want to take a moment to comment on scripting. All of the preceding interactive objects are demonstrating rules of inheritance, and those basic principles are mathematical. So in most of those objects, each student gets a different data set that are produced by the rules, and all of those objects had to be scripted. And I am not a programmer, although I did take a short workshop in BASIC at Texas Wesleyan one summer, and I did several workshops on the Linden scripting language in Second Life. But a lot of my scripts are just modifications of scripts that were written for other purposes. For example, the dihybrid garden, which pops up those red and white flowers, that is actually driven by light bulb script that was modified. And instead of turning the lights off and on, they make the flowers come up in the garden. So there are a lot of scripts that are found online, and there's a good scripting guide that you have the link for, and I will put that into the chat as well. There it is. And there are also a lot of script generating websites and YouTube tutorials. And Dodge tells me that ChatGTP can write LSL pretty well, and needs just a little bit of adjustment, and then it runs. So that's pretty cool. The next thing I want to show you is another of the interactive objects, and I separated this out from the others because I didn't write this script or even modify it. This is a script that was done by Kira Komarov, and this activity, this is the Kettlewell experiment on population genetics, and it's also at NOVA and at the Science Circle. So it's in several different, and it may be in more places than that. So it's in several places around Second Life. And this simulation is a wonderful population genetics simulation. You can pick the number of birds and the number of moths that you have, and you can actually set the probabilities, and you can set your starting populations and so forth. So this population was started with 100 moths with 75 black ones, and that's the dominant trait, and 25 brown ones, and 8 robins. And then this shows you what you got at 3 different time points there. So after 41 seconds, there were only 64 black and 36 browns. After 4 minutes, there were 18 black and 82 browns. And by 6 minutes and 41 seconds, there were only 2 black ones left and 98 browns. And one of the things you can show, there are three different backgrounds you can run here. And you can show how the backgrounds affect how the population changes. It also affects, it's different whether you're dealing with a dominant, whether you're selecting against a dominant or recessive trait and so forth. So it's just an absolutely terrific population simulation. And I recommend that, especially if you are a population geneticist, run over and give it a try sometime. So at this point, I want to see if there are any questions, and if there aren't, then we'll look at a few more things. Shada, do you have a question? Oh, in fact, there is gene mapping with corn behind the greenhouse. And I don't have a picture of that here, but behind the greenhouse, there is it shows how you do gene mapping with corn. And also, especially, how these genes interact with one another. And there are 4 different colors on those kernels, yellow, white, purple, and red, and they interact with one another. And so you can see how that works. So when you have 2 interactive genes, that's not considered a hybridization per se. That's considered interacting loci, gene loci. JC, do you have a question? Oh, what happened to the robins? Well, they're still there in the picture. If you don't stop the simulation, the robins will still continue to fly around even after all the moths are gone. But I usually don't try to starve them. I usually shut it off before then. If you run that simulation for about 10 minutes, it will probably go to stability. It will go to fixation of one of the 2 traits. But that's a good question. Any other questions at this point? Okay, well, we'll look at a few other things. We'll start with the tower. And the tower started off with, I don't know, 12 floors, I think, and now has 24. And the 2 highest floors, or next to highest floors, are now empty for whatever new projects I decide to put in there. And the tower includes most of the molecular genetics on Genome Island. And yeah, it's most of it. Just catch up here. Oh, sorry. I'm just going to skip over that. Okay. So it includes the human chromosome gallery. It includes information about gene coding and protein synthesis, and about how genes and chromosomes are structured. And there are some microbial genetics and some Drosophila genetics, a few special educational projects like the viruses that I showed you a while ago. And there's actually, way up the upper left there, you see a big platform, which is a dance floor. So you can actually have parties up there. Also in the tower, there is a blood typing activity, and a copy of that is actually over in the model educational displays that are on this island. And what this does is each of these posters is a mother of a particular type, A, B, or O, and each of them produces a set of slides, which is a possible set of progeny for that mother. And that possible progeny set depends on who the father is. And so the students will then run the blood tests and then use this chart to interpret what blood types are there in the progeny. And then from the blood types in the progeny and the blood types in the mother, they can figure out what the blood type of the father is, and then the genotypes, the genes that are carried by the mom and the dad and the children. And in for each of these mothers, there are several different progeny sets. So again, it depends on which kid is running the activity, what problems they have to solve. So this is in the tower, in the terrace that's right outside the office in the tower. And this shows a bunch of students that are showing the results of a project that they did with inheritance of genes in their own families. And they are given a pedigree kit that they can modify, so they can set up the parents and change the colors of the circles and squares, and then set up the progeny. And so they can illustrate how that trait in their family has been inherited within that family, and then they explain it and so forth. So it's kind of a fun project to do. And this is the bioinformatics platform. Bioinformatics is the use of genetic information to answer other kinds of questions, not only about the relationships among organisms, but other kinds of things as well. And several of the things that are on this platform are historical mysteries, which I'll show you more about in a minute. There is this, if you look at that little line of red dots, those are links to bioinformatics databases that students can go to. That big white chart shows relationships among different organisms that can be determined by comparing their DNA sequences or their protein sequences. And then you can see human mitochondrial DNA. It shows that. And then these circles here show mitochondrial DNA of extinct animals, and the students can compare those to living animals and see what living animal is most closely related to the extinct animal, which is on which is on the circles. And this just gives you a closer look at a couple of those. These are historical mysteries, and these are historical figures for which we have DNA sequences. And in most cases, I think we have the full genomes for all, well, perhaps not for Jefferson, but for the other three, we've got the full genomes on those that were from bones. And I usually have students do the question about Thomas Jefferson and the children of Sally Hemings. And they have the data that were the result of the offspring of the Hemings kids. And you can follow the track of the male children by looking for the Jefferson Y chromosome. So they do that as an activity, and it's sort of interesting. Oh, I forgot one thing. I can go backwards. Hold on, let me just back up a bit. There. The other thing that's on here is the role of gene duplication in producing new genes, because the red gene that allows humans and a few of the other great apes to see red is actually a duplication, copy, side-by-side copy of the green gene. And it's just modified a little bit. It's surprisingly little, but it makes a difference in the light absorbing properties of the encoded protein. So that's sort of a fun thing to look at. In addition to the eukaryotic genome galleries that I showed you a while ago, there are also a prokaryotic genome gallery so that you can see what prokaryotic genes look like. And of course, the chromosomes are much smaller, but these genomes are also to scale. That is, the big genomes have big circles and the little genomes have little circles. And each of these tells you something about the genome and about some of their genes. So here we see an example of a big one. You can practically not even see the Carcinella genome, which is a teeny tiny circle right in the middle of that big one, which is the Anabina genome. Anabina is one of the cyanobacteria, and it has a lot of genes. It's one of the larger prokaryotic genomes. So it's kind of interesting to compare those two. In the E. coli genome on the right, you can see what these genomes do. Frequently, they'll show you a picture of the organism, and they'll also tell you something about how big the genome is and how many genes it has. And this is a cell platform. And you can see the model of the cell back there in the background. And then these individual kiosks are places where you can run experiments on different aspects of cell biology. And this next picture here identifies what each of those is. So there's one for macromolecules, there's one for enzymes, there's one for osmosis, there's one for membrane transport, and so forth. And let me just catch up with the speakeasy here. And then this next slide shows me standing inside the big cell, so you can kind of see how big that cell is relative to you. And you can walk into it and walk around in it and look at all the organelles and go inside the nucleus and so forth. So the next thing I want to show you is a few student responses to questions that I asked them when I did the course evaluation. And that was, is Genome Island a game or a classroom? And these are some of the answers I got to that. One student says Genome Island is more of a classroom because when I go there, I feel like I'm stepping into a classroom. Plus the fact that the teachers there makes it seem more structured like a classroom. Another one says Genome Island is an environment that's a combination of a classroom, laboratory, and a great game. Or the labs are surrounding the learning materials that help reinforce learning. The whole island is a big game to me that I very much enjoy. Third student says, Genome Island is a new and different kind of environment. It is way more challenging than any game I have played. Not quite as formal as a classroom in a very controlled laboratory. But everybody doesn't love it. One student said, well, it's all of the above. It functions like a video game, although it's not so great for people like me who are good video game challenged. And this last picture I want to show you is a picture of the Silence in 2010. That little blue dot there shows my home place, so that is Genome now. So the northmost islands are pretty close to where we are now. And I'm hoping that somehow the Eduverse will resurrect this picture of education-related islands. And this is just a picture of the moon rise over the Abbey, which I like to look at. And we've got a few minutes if you have any more questions.
Yes, thank you very much, Max.
You
I
are
had a
very
couple of
welcome.
questions. I had a couple of questions that were sent to us in chat. And I believe you did not answer these earlier, but please let me know if you did. Shallow asked, do they do gene mapping for things like leukemia and blood disease and the probability of developing leukemia?
Actually, for the mapping or for just associating particular characteristics with the gene, there is a database called OMIM, which is Online Mendelian Genetics in Man, and I'm sorry about that, but that's what it is. And that is online, and it shows what we know about all of the identified human genes, all of the known human genes, and not all of them have been figured out, but quite a few of them have. So you can look up, and OMIM is open to the public. It is taxpayer supported, and so it is public access. and just look for omim.com, I think, or it might be org, but just look for OMIM, and you'll find the answer to about any genetics question you want to ask, and they keep it updated. OMIM. And what was the other question, Ellie?
But do you feel like students learn as much in your face-to-face classroom or more deeply? Do they enjoy it more?
I have only taught this class online, so I can't answer that question. How they would respond to the same class in a different format. But some of the students like it a lot. Uh, I think they like it better than their other online classes. I got some comments on that. They do learn it well. I mean, they do learn a lot during the course. I always do pre-tests and post-tests, and I didn't show you any of that data, but I do think they learn it well. And the interesting thing is they don't. I think it makes them more willing to think about genetics, because I've had people talk to me and say, "Gosh, now when I see a cat, I try to figure out what genes it has, whether and whether it's a male or a female." Because as you probably know, calico cats are only female, mostly. Every now and then you get a male, but mostly they're female.
And then Lucky Kane Parks has a question.
Let's see. Oh, they think it's fine. I have tried very unsuccessfully and I've given a lot of workshops and things like that, and they're always very interested in the workshops, and they enjoyed them a lot, but not a single other person at my school has moved to teaching in second line. And I'm going to try to advertise more. I took a lot of pictures this morning when I went through the Amazon, and I'm going to put some of those on my Facebook page and see if I get any response to it.
And by
So
the way,
I
his
have not
question
been
was,
successful. And even our population geneticists. I mean, I have showed him that thing, and it is such a wonderful simulation, and it would be so great for students. And I've used it with my major students, and they've enjoyed it too.
I was going to add that for purposes of our YouTube audience, the question was related to whether her colleagues received or regarded her efforts in teaching through virtual world presentations, and he asked if her work in this inspired or influenced any of her colleagues to do similar things.
Yeah, I've done several presentations. In fact, I almost signed up for one at our University College Day. I'm actually not sure I can do that, but I thought I might send in something and see if they took it. But I think I'm going to try other ways besides just presentations. Although I will certainly give everybody the link to this presentation.
Do we have other questions?
I'll tell you, the person who's been really successful at bringing other people in is Dodge. He has a lot of workshops at Nova, and he has brought, how many people have you brought in, Dodge? Lots.
Ten.
10. That's a lot. That's 10 more than I brought in.
Well, I did have another question that I put in I put it was at the very beginning.
Oh,
And
okay.
that
Yes.
was by Phil Youngblood, and he wanted to know, was that the dog calling?
Oh, very likely. Yes, anytime I get online, if there are other people speaking, the dog comes demanding treats because she knows I will give her one to get her out of my lap because she's a big dog.
So you taught her how to do that?
I'm afraid I trained her very well to how to get treats.
I know the feeling.
She's taking a nap right now, however. She's been pretty quiet for the last few minutes.
Okay, any other questions? Even related to dogs and cats?
And were there
I had
any of the other questions that you didn't get a satisfactory answer to?
You've answered mine very satisfactory, Mix. I just wondered if any of your students had stayed in Second Life after they'd come to your class in here.
A few. I have seen a few come back other semesters, but not many. In fact, one of them that I thought might come back was an education major, and she was impressed with how well it worked. But I don't think I've seen her back again after she left the class.
All right.
On the
Any
other hand,
other
if she had seen
questions?
Dodge's workshop, she might have.
Yeah, I agree with Ellie, unfortunately. All right. Well, we don't want to stop the conversation here, but we will reconvene to the social area in the back, where you may continue making observations or asking questions of Max. Really wonderful presentation. My goodness, all the effort that's gone into this. It's such a good demonstration of what can be done in a virtual world. And I wish more people could see what it was that you were doing. And my guess is that your students really appreciated the opportunity to learn in that way. We just have to spread the good news. So
Yes.
we want to give Marianne a hearty thank you for her presentation. And you are invited. You folks in the audience are invited to gather informally on the deck at the back of the venue to ask additional questions or to offer comments. Please note that the
Listen
next
just made a
installment.
great comment about old students being mentors for new students. That's a terrific idea.
Okay. Well, that's a great idea. Yeah.
It's a very good idea.
But please note that the next installment of the VWEC Expert Series will be on Friday, April 28th, 2 weeks from today, at 1 p.m. SLT. We will host a distinguished panel of educators that will discuss community building in virtual world. So this will be a panel discussion as opposed to a single person presentation. Finally, you can find links to our in-world calendar and related helpful information here at VWEC and in the Information Center. Once again, and we are available through a series of media. Once again, thank you very much for coming, and please feel free to join us at the back. Y'all.